Good afternoon, everyone. Thanks for helping us get started promptly. I think it's impressive where we are with our agenda. So I'm Althea Lang. I am a senior project manager and the portfolio lead for our Cancer Steering Committee. It's my pleasure to introduce this panel on AI applied to clinical imaging and pathology data across therapeutic areas. And with that, I will turn it over and introduce it to our moderator, Mike Montalto, who is the Vice President of Precision Medicine and Global Translational Development at Amgen. Thanks. Take it away, Mike.
Thank you. Great. Okay. Thank you so much. Welcome back, everybody. I hope everybody's caffeinated for, I think, our last panel session for the afternoon. So this has been an absolutely fantastic day so far, and I want to thank Amanda and the rest of the program committee for giving imaging and AI some airtime, some dedicated airtime. I was kind of hoping for a whole day that we could go, but we'll take the 90 minutes. So we'll try to get through this quickly and try to provide time. So in terms of how we'll go through this, I'm going to give a little bit of an overview. We're going to hear from some of our panelists as well, who will give some of their thoughts via some slides. I have some questions for them directly, which hopefully will circumvent maybe some of the questions that you guys have here in the audience, and we'll try to provide some time for question and answer as well. So let's see. Let me get started here. So imaging, pathology. I put this slide in at the last minute. Thanks again, Amanda, for that this morning. Just to do two things. One is to give a perspective of biomarkers across the drug development continuum and the different places and phases. We've had a lot of discussion today about the different kinds of biomarkers that exist. Of course, there's the best category, but we layer these on in drug development into the various phases of drug development where some biomarkers are more important than others throughout that continuum, and particularly imaging and pathology have played a very critical role in the drug development process. And I'm just highlighting a couple of examples. There are many around pathology and imaging, but of course IHC is a workhorse these days and has been for a long time in oncology in terms of identifying patients. We heard about that a little bit today for high biomarker, low biomarker, and how that can be predictive of response, particularly against various targets. And imaging is a very important biomarker, surrogate biomarker, so to speak, in progression-free survival and best overall response in many of our cancer trials. And there are many other examples of where imaging plays roles. And of course, we've heard about bone density as being one of the very important ones recently. Interestingly, though, we'll find that almost all imaging that's done today, as we think about imaging today, as we all sit here together, does not employ AI. And yet imaging and AI are becoming very intimately linked for reasons that I'll talk about. So artificial intelligence, this slide is already not totally relevant in a way. So this is just giving an overview of how AI works, essentially, where we can sort of take various data inputs. Let's see if this works for pointer-wise. But we can take various imaging inputs on the left, whether it's input data, imaging data, NGS-based data, put them into various machine learning models and make predictions. At the end of the day, that's what we're trying to do. Much of the data that we use as inputs for the prediction of AI models comes from images, and that's sort of what I'm highlighting here on the right, comes from images in pathology, particularly as it relates to linkage to clinical data, which puts imaging and pathology kind of in a unique category for the amount of data that's available linked to clinical data over time versus other particular modalities. So it has those unique attributes. So when we talk about just imaging itself, imaging and pathology have these unique characteristics. As I said, they're available in routine clinical practice and have been for many decades. The spatial context of the biology matters significantly. There has been an eternal debate between grind and find versus seeing is believing. I'm in the seeing and believing camp. They're both very important, and neither one of them will ever win out, but particularly in the day and age now of multimodal. But spatial context we know does matter. The orders of magnitude of data that's available in images is intense. The amount of data that one can get from a single image, whether it's pathology or radiology-based imaging, is vast. We can always go back to the original imaging source. I think that's an interesting thought. As new emerging platforms are coming out, we talked about earlier plasma-based epigenomics, How you collect that data is important and it's relevant to the test. In the imaging world, there may be new models that are developed over time. We can always go back to the original images and apply those models to see whether or not they're making improvements. Whole slide, whole organ scale minimizes sampling bias. It does not eliminate sampling bias, but in today's world where radiologists or pathologists are sampling where on a slide or where on an image to do their analysis, we can circumvent that looking at the entirety of a whole slide or whole image. Readers themselves provide quality oversight and interpretability. Interpretability is very important. I'm sure we'll talk about that. But the fact that trained physicians oftentimes are overseeing and understanding these images provides a certain level of oversight that we don't necessarily have in other biomarker platforms. And the quantitative scalable outputs themselves can be somewhat more accurate depending on what accuracy is and what your outputs are. These so-called sort of human impossible tasks, there's a lot that we can do there. So AI has particular advantages, I think, in the context of pathology and imaging as it relates also to specifically to drug development, which I'll talk about here. So I have about 5. There are many actually challenges associated, I think, with pathology and imaging as it relates to drug development. One of the biggest challenges that we have in traditional imaging and pathology is the variability and subjectivity that inherently exists as we try to read these images as a human reads them and tries to determine what they are seeing essentially and sort of aligning on that. So we see a lot of reader variability. That leads to the second huge challenge, which is adjudications. So when 2 readers or more than 2 readers disagree with what they see in an image, we have to figure out who is right and how we determine who is right. So in the case of adjudications, it adds a lot of time and expense, and it's not scalable. And that's a huge consideration when we're dealing with how to use images in drug development. And of course, you can see on the slide already, and I won't drain the whole thing, but on the right, every single one of these things, artificial intelligence can mitigate substantially. The data volume is huge. It's nearly impossible for a human to get through the totality of the data volumes that exist in images, and so AI can circumvent that in a very expedient way. Lack of sensitivity and reproducibility of quantitative endpoints is a huge deal, and that can translate into early missed endpoints. And so we can mitigate that through AI. And just as I said, the biological heterogeneity that exists can be mitigated using AI on top of imaging. So it's hard for me to think about imaging in the future without having AI as a component to imaging. And like I said, as we sit here today, imaging is not necessarily adopted yet, or AI is not necessarily adopted yet as a component of imaging in practice, both in drug development as well as in clinical practice yet. It certainly is on its way. Why is that? This will be my last slide. We're very fortunate to have one of our panelists who co-authored this paper, Chi Liu, and also I think helped co-author some of the most recent draft guidance and the considerations for use of artificial intelligence. But some of the challenges that exist in using AI in drug development are sort of limited here. So limited data access, data is very important. As we talked about today, data consortiums, sharing data, making data available for us to both train models and validate models is a critical component, and it can be a huge bottleneck in developing models. Confidence in the interpretability of what we see in our models is very important and can be very challenging and can slow down the development of these. The regulatory complexities are somewhat vast. As AI is moving very quickly, keeping up with the pace of that from a regulatory policy perspective is challenging. This draft guidance, by the way, does a great job. If you haven't read this, I would read it, in starting to address and starting to provide a very good framework for understanding what is the context of use, starting there, understanding what your risk is, and understanding what your evidence needs to be in relation to that risk. So this is a really fantastic step forward, and perhaps we can talk a little bit about that today as well. And the siloed disciplines, because AI and the multiple disciplines that are involved, getting everybody to sort of be conversant in the various different technologies that are relevant can also be a huge challenge for us. So these are the things that hopefully we will get an opportunity to address as we go through our panel, and that hopefully will set the stage. I think we're going to go a little bit in this order: Jeff, Andy, and I think Steve and Susan, and then Chi is going to give a little bit of a talk. Okay? Awesome. Thanks. And I will ask Jeff then to come up here and give his perspectives. Jeff is the president and CEO of Friends of Cancer Research.
Thank you, Mike, and thanks to the Biomarker Consortium for the chance to share a couple of case examples of two projects that we've been working on that hopefully can be examples of how multi-stakeholder collaboration can hopefully accelerate the understanding of different technologies and ultimately their implementation. The first is related to Mike's comments around the potential role that AI can play in digital pathology. Given the wide variety of opportunities for AI-enabled tools in this particular space, we wanted to select one particular use case that we could sort of dive into. In this case, and in many other cases, there are multiple different technologies that are coming online that have very similar contexts of use, but perhaps not the opportunity to better understand how their outputs relate to one another. So in this particular pilot, we partnered with a large group of people in order to bring together, in this case, 10 different HER2 measurement tools that leverage AI technologies to evaluate digitized scans. Our collaborators from a hospital in Belgium were provided access to 1,100 digitized breast cancer samples in which our data partners evaluated the HER2 expression. This has been a long time important biomarker in breast cancer and emerging in other cancers as well. But in recent years, the reclassification based on HER2 expression through the development of antibody drug conjugates has sort of regrouped breast cancer and identified particularly promising compounds for low HER2 expressors, which in years past would not have had access to these types of therapies. So there's a great deal of potential for the AI-enabled digital pathology tools to detect expression of HER2 in ways that perhaps human pathologists themselves may not be able to readily do. So what you can see in this slide here is across each of the individual columns here are the over 1,000 different samples. And then each row representing a different digital pathology HER2 evaluation tool. And then those denoted P1, 2, and 3 are human pathology reads. So you can cross-compare the outputs by the different tools and compare them to the human pathologist read. The sort of sum here is that on the right-hand side, the dark blue are the high expressions of HER2 3+, and you see great agreement across the different tools here. And similar than the 2+, relative agreement across the different tools in the human assessors. Now, as you go to the left and HER2 expression becomes lower and lower all the way down to those that had 0 expression, is where you start to see some level of variability. So even the opportunity to understand the differences of the outputs of tools that have a similar context of use, I think is an important component of understanding their applicability, particularly for these types of tools that have different data sources that they're being trained on in order to construct their algorithms, where a typical validation study for each individual tool may not be able to recognize the potential for variability here. And given the level of agreement here, in this case, it's if you look across all expression levels, it's almost 80% of agreement. So hopefully understanding performance of these different tools will help increase their confidence in thinking about where they can be applied. In a similar pilot that we are currently constructing now, trying to follow the same sort of framework of cross-comparison of different tools, this is a project that, again, builds on some of the things that Mike was describing around the potential role of automating tumor assessment in order to minimize bias and increase efficiencies in clinical trials. What we've constructed here is partnering with about a dozen different tool developers that have the capability to measure tumor change through AI-enabled tools, and have identified a common data set which each of the partners will evaluate and will be able to understand the extent to which they agree. So the purpose here isn't just to find scenarios where variability exists and become concerned about it. It obviously has to be clinically meaningful in order to understand where that variability can be minimized and what factors can be put into future tool development in order to minimize unnecessary variability, but again, trying to create confidence in the application of these tools. I think Mike mentioned this in his summary of application of AI in imaging. With the current paradigm for clinical trials that are used for registration having to go through an adjudication process in order to ensure that, or at least minimize the potential for local assessment bias. But having an automated tool that could help with the adjudication process could save months and millions off of clinical development programs if we can get to the point where we are able to increase the confidence and reliability on these tools. And perhaps these cross-evaluation types of studies can be a component of that by transparently demonstrating the performance of these different tools. So I look forward to the discussion, but thanks for the chance to be here.
Great. So thanks for inviting me, and really excited to be part of this panel. And I'll be presenting quickly on AI-powered pathology for precision medicine. And just to provide some context, and really building off both previous talks, there's a tremendous opportunity in imaging broadly, including radiology. At PathAI, we focus on pathology to build a single platform powered by data sets that grow larger year over year, and where there's a core foundational set of deep learning models to do what any single pathologist couldn't do, which is to interpret essentially every cell on every slide across every therapeutic indication where we're taking out tissue. And it's such a critical area because this is one of the few non-tissue destructive new technologies where you're actually preserving tissue. If you think about the standard of care in clinical diagnostics and historically entirely in drug development, these images weren't even captured. So the tissue is removed from the patient, it's processed into a block, a thin section is cut out, and the image may exist very, very transiently in the brain of the pathologist, and then it's gone and disappears and can't be used for knowledge and advancing either field. So now with deep learning, we can have models that will improve year over year, and then deploy the same technology in drug development as indicated on the left and in clinical diagnostics indicated on the right. And essentially everything a pathologist is trying to do is to make the right diagnosis and to score the correct biomarkers to best predict both prognosis as well as response to treatment. So biomarkers are really the core of pathology. And just to quickly go through the area on the left in drug development, we build models that could be used for exploratory research. And we would hope that in every clinical trial, you have models that are used to generate more and more information about which features in pathology predict response and resistance. But also we build highly regulated tools that will be used to support pathologists in enrolling patients in trials and assessing histologic endpoints. And we also partner with pharma partners on really the next stage of regulated biomarker here, which would be a companion diagnostic to be used not only in a registrational trial, but also to really support patients in the real world for identifying patients who will show the safety efficacy profile for newly approved therapies. And to really realize that real world, you have to not only have the platform that's ubiquitous on the left, but you also have to be available in labs around the world so that you can really accelerate the speed at which new insights with drug development can be disseminated broadly globally. And this would be impossible to do when relying on humans, but it actually is very feasible to do when you have a cloud-based technology that's available globally. So that's really the mission. And to give you a couple of recent examples, so for the past 5 or 6 years, we've been working on a tool. Initially, we worked on it so long that the disease even changed names during the course that we were working on this. So this was submitted to the Biomarker Qualification Program as our AIM-NASH tool. It's now AIM-MASH, which is the first and only AI tool qualified by both the EMA and the FDA for assisting pathologists for enrolling MASH clinical trials, as well as scoring histologic endpoints. And we received the EMA qualification in March of 2025, and we just achieved the FDA qualification in December 2025. So we think this is the first AI tool, certainly in MASH, that's been qualified by both the EMA and the FDA. So this was a really rigorous process of both analytic validation and clinical validation. We published 2 different papers in Nature Medicine, one on the analytic development and a second on focusing on the clinical validation. And we think this can really be an important new paradigm for future ways of incorporating AI assistance into trials, particularly in areas, and MASH scoring is certainly one of them, where it has tremendous impact on how trials are executed, tremendous impact on things like response rate, and also there's tons of variability between pathologists because the tasks that are in the scoring criteria, I would, to some degree, put in the category of pathologist impossible to what Mike said, things like counting every ballooning hepatocyte in a slide when there could be 100,000 cells in that slide and people struggle to agree on even a single one. That's obviously an impossible task for humans, but it's a very feasible task for an AI system. So that was one big advance from last year. And a second disease area which shares a lot of the similarities in terms of challenges of pathology include inflammatory bowel disease, which has been another major area of focus outside of oncology, where we've been partnering with FNIH to bring reproducible, standardized scoring of IBD as well as histologic healing to UC trials. And really just to show this full spectrum of both using the exact same image for exploratory translational research and biomarkers on the right, but also building more regulated and focused algorithms to support the scoring of tasks pathologists are already trying to do today on the left, which is our AIM AI-based measurement of histologic improvement on the left for ulcerative colitis. So that's our AI tool to support the reproducibility of endpoint measurement and assessment of therapeutic efficacy with AI assistance. So this is a very specific readout of a very specific set of scoring criteria. And then our much more broad and exploratory tool on the right, IBD Explorer, which measures essentially entirely sort of pathologist impossible quantitative features and capturing over 1,000 spatial histopathology features at the cellular level to power both new biomarker discovery as well as insights into mechanism of action. And of course, these two things can really work well together for defining future biomarkers. And at PathAI, we focused significantly on not just building the AI component, which is very important, but embedding it within a GCP, GCLP compliant platform and even our end-to-end lab that we have in Memphis, Tennessee, so that we can support prospective clinical trials all the way from phase 1 to phase 3. So we think there's tremendous potential for all the reasons already mentioned, the ubiquity of the data, how rich the data is, and how limited just relying on 1 to 3 pathologists to score it is in terms of both time, cost, as well as just data quality. So with that, I'll conclude and hand it over to Stephen.
Thanks, Andy. First, I want to thank the Biomarker Consortium and congratulate them on 20 years of very impactful research and hopefully looking forward to the next 20. But also want to, sort of look ahead, as Andy nicely set up, what is the future of using AI tools in clinical trials? And so recently, I've had the privilege of serving as the co-chair for the Mucosal Healing Project Group, along with my colleague Russell McBride from Mount Sinai. And that particular group had a couple of aims, very, very robust and specific aims. One that we won't talk about here today was to address an issue that we've long known as a problem in IBD, which is sampling bias. And the group at Mount Sinai looked at how many samples does it really take from a colonic biopsies to actually get an accurate assessment of the disease burden. And that's not an easy question because in a lot of clinical trials, a lot of clinical readouts in IBD, you rely on a piece of tissue that's no more than 3 to 4 millimeters in diameter, and you're expected to extrapolate the entire disease burden for the colon. That's not an easy thing to do. But the second major aim that we want to talk about here today is an extension of what Andy just set up. So the Mucosal Healing Group set out to develop a tool or tools that would be able to not only help us get an accurate, reproducible, and robust assessment of histological disease, but also to extend that into more exploratory areas to develop a way of looking at features that are underappreciated or cannot be appreciated by the human eye, but yet will inform on disease. And so what happened was the Mucosal Healing Group, we partnered with PathAI to evaluate the two tools that Andy just talked about. AIM High UC, which actually is meant to score in an existing metric, the Gabos, RHI, or Nancy scores, and recapitulate what central readers will do in a clinical trial, versus the IBD Explorer, which, as he said, is a feature extraction tool which captures novel biology. So what you can see in the centerpiece here is sort of a very brief condensed version of what we found in that. More details are actually going to be presented in a poster today by Autumn Jones, who is currently hiding in the far left over here. She thinks I don't see her, but I do. She's right over there. I encourage all of you to go by and look at that poster. It has a lot of really good details about this project, which I simply don't have time to cover right now. The upshot of it is that when we looked at AIM High UC, and this was truly a group effort, the consortium partners all contributed data sets from their IBD clinical trials across multiple modalities, multiple MOAs. And what this allowed us to do was to show that the AIM High UC tool was either non-inferior or in some cases superior to individual readers compared to a reference standard for grade level Gabos, RHI, for most key endpoints that we typically look at that are histology anchored in IBD clinical trials. And so that was really good. We thought that was great. The IBD and AIM High UC explorers, as Andy said, are actually ready for prime time. They are on compliant AI clinical trials platforms and are ready to be used prospectively into clinical trials. So that being said, we should be doing cartwheels. It should be adopted, by next Tuesday, and everybody should have a standardized way of looking at things across the board in IBD. Here comes the other side of the coin. And, Janet's, Dr. Woodcock has illustrated some of this earlier this morning, but there are major concerns, reservations about widespread adoption of these types of tools in clinical trials and in drug development in general. They're very complex, but I try to distill them down to 4 major areas: development, deployment, acceptance, and use. Now, each one of those topics could merit an entire panel in and of itself, but what I try to do is distill this down into some of the more pertinent, pressing concerns. With regard to development and training, when is good agreement good enough? Dr. Woodcock, you said that's actually not really our goal. We really don't want good agreement with what the existing gold standard is today. We want something better. However, in taking that leap, that sort of, quantitative leap to the next thing that's going to be better, we run into some hurdles and roadblocks about acceptance, which we'll talk about in a little bit. So we try to take baby steps. We wanted something that does really well on what we already know and what we already accept. But now, good agreement, good enough, the more provocative question is something I think Stephen brought up in the last section, is that how do you get around training a tool and training bias into that tool because you're relying on humans as your ground truth? And one of the things we as the Mucosal Healing Project team quickly realize is that in the not too distant future, when does a tool become a better ground truth than what we currently have with humans? Now, albeit they're highly trained humans, but even in a consensus, even in a group, you still have either individual bias or group bias. So you'll never get 100% alignment. So when is good agreement good enough? And when does the tool become the better ground truth? This is something we struggle with. I'm sure all of you can appreciate. It's a very complex question, and it's something that we hope will spark more debate. As far as deployment, now in clinical trials, this is also very important. What weight do we give an AI finding for a given endpoint? How much do we trust that that's actually not going to alter in a significant way the findings of a clinical trial? So what weight do we give to that endpoint? And when does that become decision-making, not only in the process of drug development, clinical trial assessment, but also patient management? When does that tool become decision-making? Also a very complex question. With regard to acceptance, now all of those uncertainties I just spoke about lead to a problem with acceptance. The roadmap for regulatory approval is not as well defined as we would like it to be. Part of that, at least from our perspective, is that it depends on what you want the tool to do. If you want the tool to just basically speed up your reading faster, that's not such a hard lift for regulatory acceptance. But if you want it to actually become a novel endpoint, a surrogate endpoint, a digital endpoint to assess disease burden, that's an entirely different conversation as far as regulators go. And then you have to get alignment across the different agencies upon what they expect. And that's not always the case either. What FDA views as important may not fit with EMA's vision of what needs to be done. The second one is the real kicker. And this is what makes a lot of people nervous. What happens when the tool gets it wrong? Now, in the inflammatory space, is not as critical, but if you go back to oncology. Now, if the tool gets it wrong in oncology, there are real-world consequences. And if it does get it wrong, who's to blame? So these are some of the uncertainties that we hear frequently, not only from regulatory representatives, but also from our KOLs, from our key opinion leaders. They get a little nervous about this. If I'm truly going to treat my patient based on this tool, and especially if it's an unsupervised tool, a black box tool, there's a lot of voodoo involved, and they're not comfortable with that, making a treatment decision based off of that. Then we come to use. Now, who will have access to it? Andy touched upon this also as well. Do we have the ability to have everyone have access to this tool, or is it just specifically for clinical trials? And does your gastro in a very rural setting have the same access to these tools, and will they use them? You know, it's education as much as it is anything, as much as it is data, it's education. Will they use them? Will they have access to them? And then how likely are they going to make treatment decisions based on those findings? That is the end game. How will they manage their patient based on the findings of these tools? And so with that, I'm going to conclude. Those are a few provocative thoughts, some concerns that hopefully will spur some discussion in the Q&A. And now I'm going to hand you off to Susan, who's going to give us her views on how AI fits into the sort of the biomedical space. Thank you.
Thank you so much. It's a real pleasure to be here, and I want to thank FNIH for their many partnerships over the years for the Biomarker Consortium, but also for the AMP projects. Wonderful. And then for some of the work we're doing on real-world data, significant opportunities with FNIH, and glad to see our colleagues here. So I'll just tell you a little bit about some opportunities in AI at NIH. So we know that there are a lot of important considerations for biomedical AI. For example, the need for innovation and data fusion and model training, just like we were just talking about, as well as model assessment and then the downstream applications. These are things that are front and foremost on our minds as we're starting to develop new programs. We want to make sure that our solutions that are innovative also have sound approaches and are consideration for our stakeholders. So I often talk about co-design, which is you develop algorithms and tools with the patients or the communities that you have in mind. And that's really bringing everybody together at the table at the start of the program. And I think that's something that FNIH is really particularly good at. AI researchers, patients, biomedical scientists, and healthcare communities really do need to be in the loop as we start to think about AI systems. And I'll talk a little bit about primed AI and multimodal AI. AI is really a fast evolving field. There's a lot of really innovative work that happens every day. That work is not always commensurate with the NIH timelines for funding. That's just how it is. We need and we try very hard to be flexible and agile in our approaches to development. And that means we have to go outside of the box, and that's hard too. So these are some of the challenges that we face and that I hope that you'll help us solve. I wanted to bring up Bridge to AI, and this is sort of the data generation project that happened for the last 5 years. And there are 4 main grand challenges in precision public health. That's mostly looking at work on, oh gosh, this is the one I actually forgot the details of. Critical care, this is in the EHR, looking at how we can use real-world and healthcare data in AI. Salutogenesis is how to understand going back to health from disease. It's a fascinating program working here in type 2 diabetes. And then functional genomics is cellular functional biology and all of the multi-omics data that you get from studying cells. I bring this up because 2 weeks ago we approved the second phase of Bridge to AI. And so we will take these data generation projects that are our flagship projects with very strong, robust AI-enabled data, as well as methodologies and best practices, and now turn this into a challenge and a funnel for researchers to take these data and then develop tools, methodologies, applications, devices through a funnel approach, not too dissimilar from what you might have heard of during COVID, the Shark Tank approach, where you start with a sort of an idea and then you go from idea to data, to generation of algorithm and methodology. And finally, we hope to actually get to FDA approval for methodologies, for devices, for tools. So this is something that you'll start to see coming in the new year, the opportunity to really leverage the work that we did in the first 5 years of Bridge to AI. Super excited about this. We have been working on opportunities to advance health research through multimodal AI, sponsored by my office, but also now expanding. This is an opportunity to bring biomedical researchers to engage with patients and clinicians to develop multimodal AI capabilities. We are looking at system-level challenges across diseases, also engaging ethical researchers in the idea of co-design with clinicians. This is something that we're excited to also partner with our Chief AI Officer as we expand this program for the next year. These are just some of the awards that we've made across the United States and in their focused area of research, including breast cancer, looking at predictive outcomes for critically ill patients, diseases and phenotypic discovery systems, lupus, something that we're passionate about. So some of the work that we've been doing over the past 2 years, and I'm hoping that we'll continue multimodal AI in this next program, which is a primed AI common fund program. Across the institute centers and offices, we're partnering on looking at how we can leverage clinical imaging. We know that it plays a really important role in healthcare. Often it's isolated from other types of healthcare data or even other types of real-world adjacent data like genomics, proteomics, multibiomics. We want to integrate these different types of data together. So this is an opportunity in the future to leverage clinical imaging, integrate in other types of data such as genomics data with the prime goal of accelerating clinical decision support methodologies and tools for clinicians. So this is an idea where we're going to bring clinicians, patients, and researchers together to help develop these tools that will be enabled through clinical practice. You should look for these announcements. I've been waiting for a little while for them to come out, but these funding announcements should be out this spring. And it's a really great opportunity to partner in public-private partnerships is what we're actually looking for here to really enable clinical decision support tools focusing on clinical imaging. So the real focus here is on those imaging data sets. And with that, I am going to pass to Shu, who is going to tell you a little bit about the FDA's drug development and regulation. Thank you.
Thanks, Susan, and good afternoon, everyone. Thank you for the opportunity to share my views on the application of AI in drug development and regulation. I would like to start with first painting a picture of the regulatory landscape for AI-related submissions at CDER FDA. In the figure on this slide, you will see the number of regulatory submissions with AI or machine learning-related components from 2016 to 2025. As you can see, before year 2018, there were actually very few regulatory submissions at CDER that has AI or machine learning components, maybe 1 or 2 submissions per year. Then between 2018 to 2020, there's about 2 to 3-fold increase each year. And then in 2021, the numbers had a very sharp increase to over 100, which is about 10 times higher than the year before. And then from 2021 to 2025, you can see a steady increase of the number of such submissions each year. And personally, I believe this trend of increasing applications of AI and machine learning drug development is likely to continue. And in this landscape analysis, we also summarized the why and how the AI application was used in drug development. The box on the top shows the type of analysis in those AI applications. And I'm not going to go through everything, but I want to show you that, sorry, I have to go back. Imaging AI was frequently used for imaging data analysis. And also on the lower half of this box, you can see endpoint biomarker assessment was often used when AI was used for drug development. Actually, based on our new analysis, this seemed to be the biggest category, like highest number of application of AI is in this category. And another point I want to make is the objectives here listed are not mutually exclusive. So AI used for biomarker application, those can also be used for enrichment trial design, patient risk stratification, or dose selection, etc. And FDA has been very proactive and active when it comes to AI. In this slide, we summarize the AI-related initiatives at CDER. And this includes a series of landscape analysis, a series of workshops, discussion papers, and also the development of draft guidance on the use of AI for regulatory decision-making. In January last year, FDA published draft guidance on the considerations for the use of AI for regulatory decision-making in drug or biological product development. And this draft guidance provides a risk-based framework for establishing and evaluating the credibility of AI in AI application in regulatory decision-making. And we introduced this concept called model risk. And this model risk is driven by two factors: model influence, which means what is the weight of the model output for your decision-making, or in other words, what other information will you be used for the decision-making in addition to the AI output. And another dimension is decision consequences, which means what will be the consequences if we make the wrong decision. And either an increase in model influence or decision consequences can increase the model risk. And the model risk will determine the validation activity that is expected, the acceptance criteria, and also what kind of documentation will be needed for the AI use in drug development. Looking ahead, we still have a lot to do. We will continue to seek feedback from all stakeholders, and we will continue our responsive, risk-based regulatory approach. And we will try to advance science, regulatory science through innovation and collaboration. And we look forward to continue this joint journey with all of you. With that, I will turn back to Mike for the panel discussion. Thank you.
There we go. That was fantastic. Thank you all so much for your slides and your prepared remarks. And I think that was a very good overview and give us multiple perspectives. I think so. I'm going to seed with a few questions and give people a little bit of time to think about some of the questions they have. So my first one is, Stephen, for you in relation to kind of the industry perspective. So in your opinion, and sort of speaking on behalf of the industry to some extent, where do you see the biggest opportunities for using AI in imaging and drug development, and where do you feel like the industry is currently on that journey in terms of their willingness to adopt it into the context of their trials?
Well, that's a great question. I think, again, speaking from a gastro perspective, one of the opportunities I think for AI-generated image interpretation is probably in the screening and enrollment of a clinical trial. One of the things we struggle with, and one of the things we have to do better at, is to cut down our screen fail rates. And in gastro, typically that means they have signs and symptoms, but they may fail their endoscopy. Case in point, with the emergence of sort of non-invasive technologies like intestinal ultrasound, we are hearing across the board, and we have an initiative currently to say, bring the patient in. This is an example. Bring the patient in, give them a 15-minute intestinal ultrasound. If you can't find pathology with an ultrasound, do not send them off for that endoscopy. Do not have them go through the bowel prep, the patient burden, waste their time. If you are not confident or 100% sure that you have pathology that you can observe with a non-invasive measure, and they're going to screen fail you anyway. So this is better for the patient. It's also better for streamlining and making clinical trials more efficient. It helps build more robust data if you can cut down that screen fail rate. I think that's first and foremost where the field is headed Now that we have access to more non-invasive techniques to do this, I think picking the right patients for your trial is key. You know, other opportunities for imaging where they can make a big impact is a thing that Andy and I have talked about as well. If you can take things like histology and layer that in with sort of spatial data, and you have an AI predictive model that can help you figure out what patient endotypes are present, does that now help you with the sequencing of your drugs for this patient? And as important as as much as we place importance on predictors of response, equally important to us are predictors of non-response. So if you can identify a set of features that gives you confidence that that patient is unlikely to respond to an anti-whatever, how do you integrate that, and how do you help inform treatment decisions about how you sequence a subject on drugs? I think those are probably two key areas where imaging data could play a big role.
Excellent. Thank you. Okay, so my next question is for you, Andy. We'll sort of stay a little bit on the sponsor industry perspective to some extent. So congratulations to you and your team for getting the first AI approved or biomarker qualified for pathology. What did you see? So we heard earlier today as well from Dr. Woodcock that there are huge challenges in just bringing biomarkers forward and getting them qualified. Data, of course, is important, and it is scientifically very rigorous to meet a high regulatory standard. And those of us who have been through devices at CDRH understand that well. The biomarker qualification program is somewhat different. So what did you see sort of firsthand in terms of some of the challenges going through the qualification process, and what sorts of recommendations might you make to improve it? Yeah.
Sure. Thanks, Mike. So I can talk about some of the things that we did that made it much easier than it might have been. One was starting with already sort of FDA recommended criteria. So we did not need to develop and then validate novel new surrogate histologic endpoint criteria, which would have potentially made the thing 10 years longer. Who knows? Because then you have to show that it predicts outcome as well as what's already been recommended. So starting with what's known was really important, and I think to Dr. Woodcock's point about going incrementally, like we think that's absolutely the first point. This is how trials are being enrolled today. However, it's well established that there's tons of variability within and across pathologists, which is you can do a very simple statistical or simulation analysis to show how this is increasing noise, increasing in certain cases screen fail rates, and then for a truly effective therapy, making sponsors' power trials much too large to deal with all that noise, and increasing the type 2 error rate, which would be a trial failing to hit its endpoint despite being effective. So like for all those reasons, we thought let's start with sort of a better thermometer to measure the biomarker that's already recommended. So I'd just say that made our AV, analytic validation, clinical validation path much more straightforward. And I think it was a great process in the sense of the scientific feedback and the design working with both the FDA and the EMA in lockstep. And that went really well. I think it was a really well-defined process going from a letter of intent, an LOI, to a qualification plan, to a final qualification plan. And we also sort of had a pre-competitive type of consortium with multiple pharma sponsors, which helped us tremendously, like Michelle from Novo Nordisk and others in the room, for enabling the data set and just the expertise to ensure we designed that correctly. And I think all that went well and was a really good role model. I would say the one thing that was legitimately challenging was just how unpredictable the reviewing timelines were. So all of these things are both costly and take time, but those were known entities. But unlike things like a typical medical device, which has the medical device user fee amendment, where there's an agreement, an additional fee paid by the industry sponsor of the medical device to get it in, say, the 510K process, but at least you have a predictable timeline. And similarly for a drug, there's PDUFA. There simply isn't that here. And I think it was very difficult for the FDA to prioritize these submissions. So it just took much longer than we expected. And like, for example, the letter of intent took a couple extra months. The review of the qualification plan took essentially 3 years versus 6 months. And the final qualification plan took about 18 months versus 10 months. And we realize there's a huge amount of work to doing this well, but it's more like, is there a way, and there should be, I think, to creating a system where those timelines are expected and the industry sponsor can factor that into the decision of should I just go for, in this case, the 510K, or should I do the biomarker qualification plan? Because the context of use was perfect for the BQP, but the timeline would have been more than probably twice as fast if we'd done 510K, and it's not clear sponsors would have really noticed a major difference.
Excellent. Thank you for that. So, Chi, this is for you. So we'll head right now into the sort of regulatory, a little bit perspective. So, and perhaps I think the guidance may address some of this, but what do you think, in your opinion, sponsors kind of most commonly maybe misunderstand about the agency's expectations when going through a qualification process for an AI-based tool?
That's a great question. Oh. Thank you. I think one thing I have observed in many submissions is sometimes sponsor would assume a good model performance will automatically translate into regulatory acceptance, which may not always be true because the standards depends on the context of use. And very often a lot of sponsors do not specify the context of use that clearly. And as we talked about early, FDA used this risk-based credibility framework. So when we decide what will be the regulatory acceptance criteria and what kind of documentation we need to see, really depends on the model risk. For example, when we talk about imaging biomarker, an imaging biomarker can be used for patient enrollment, endpoint assessment, or patient stratification, all of them associated with different model risk. There will be different expectations. And in addition to that, when are you going to use this biomarker also matters. Are you going to use in phase 2 trial or phase 3 trial? Are you going to use it as primary efficacy endpoint or secondary endpoint? That also change the standards or exploratory endpoint. And also what kind of guardrails do you have? Also change how we view the acceptance of this whole package. For example, whether there will be human in the loop, what will be the QC plan, and what will be the consequences if the model fails. All of this will need to be very clearly specified in the regulatory submission, and that can really facilitate a faster turnover. For many of the DDT or IND submission, sometimes I find a lot of time was taken because there has to be so many IR or information requests. So whenever there's back and forth of information request, and very often we got the response of the information request, it generates more questions. So that really prolongs the timeline. So I think if in the future when there's more examples out there and people know what exactly the agency will be looking at, will be requiring, that will help the communication and make the process more efficient.
Awesome. Great perspective. Okay, so we'll stay on the FDA for Brandon for your question, and I didn't get an opportunity to introduce Brandon, but Brandon Gallis is a research physicist and mathematician with the FDA at CDRH in the Office of Science and Engineering. And I would say personally, a world's expert in understanding sort of technical aspects of verification and validation as it relates to imaging and pathology. So Brandon, in your perspective, what tools or infrastructure gaps are kind of slowing right now the regulatory confidence in some of these submission packages that are coming forward?
Thanks, and I'm really happy to be here, even though I didn't have slides. Life is still a little bit tough where I'm coming from to get clearance for that kind of thing, but I really appreciate being invited and all these panelists, happy to share the stage with them as well. And this is my second time speaking at an FNIH workshop and the last time was to introduce digital pathology to this community as an idea for a project to go forward. The community here wanted to see some things in digital pathology and AI in their portfolio because, if you think about who makes the cancer decision diagnosis, it comes from a slide 99% of the time. And so now that we are saving these slides as images digitally, and we can recall them at any time, we can include them with other data, the opportunities for for discovery as well as speeding the practice of medicine. And so I'm from the Center for Devices, and I'm in the Office of Science and Engineering Labs, which is the research arm of the Center for Devices. And so I wear two hats. One is to do research, and the other is to do submission reviews. And I've been there 24 years, so I was there when mammography went digital, and that was a big deal because we didn't have the methodologies for evaluating digital mammography. You know, it was a beauty contest of this image looks good and this image looks good, and the performance got all muddled when you tried to compare the performance with screen film to that with digital mammography. And so the real key that unlocked that was statistics. And that's what I've been focusing on in my tenure at the FDA, is improving the statistical methodology and study designs, specifically involving the human. So we've heard already talked about the reference standard coming from the human. It's fraught. You know, you can't get the same answer from two radiologists or two pathologists or the same pathologist on a different day. And so what do? We average them, and then we close our eyes to that variability. So we used it for the readers' variability that are using the devices to decorrelate and account for those variabilities, so that the error bars that we get from performance assessment generalize to new readers as well as new cases. We're always understanding how to run a study. We need multiple cases, right? But we also need multiple readers so that we understand that this will generalize not just to new patients, but to new readers. And so my current research has been to flip the script a little bit, because now we are using readers so much as the reference standard. And that is not the same problem as being on the front end. And why? Well, I'll tell you, it's all about 1 over n. The truth defines the positive cases and the negative cases. And when you do your sensitivity or specificity, you have a 1 over n. Now that n comes from the truth. And if that truth is variable, you have a nonlinear entry into the statistical methodology. So that's what my research has been about lately, is trying to derive the metrics. I've hosted and led a few studies with hundreds of patients and multiple pathologists per patient to try and understand that variability, try and build that into the metric, the performance metric. So now when we have a performance endpoint, we can think that the error bars will generalize to new truthers, so that we don't have to worry about some arbitrary adjudication, like we like this person's answer the best because they've been here the longest, and if I don't choose them, they'll be angry with me. And those sorts of things, to turn it into a statistical problem, are within reach, but it does take more effort in understanding the reader and collecting that multiple reader data and spending the time. One of the biggest things that I see in studies at the FDA that come to us in submissions is, how did you train, or what instructions did you give your reference standard readers? And then, they just did what their clinical practice is. And then the question I get from my studies is that good enough? And so my question for device manufacturers, is that good enough? And the thing that I like to collect is, well, show me the variability of your reference standard. And when they do, the light bulb goes on for everybody. That is just not that simple. You need to have strategies to have multiple readers, to bring them together. And it's not always on the consensus side to try and get a single truth, but it might be to treat each expert as a noisy version of the truth, and then average on the performance metrics that you get from each one of those, because that statistically is a much easier problem. So when I talk about gaps, those are the gaps that are really going to help us, because the early studies in AI are all being validated by a reference standard expert panel. Nobody in devices is willing to wait for outcomes. And so that certainly is a much stronger endpoint, and I love it. But I never see it in my reviews. So those are the things that I think about gaps. And the next solution to those sorts of gaps is community-based data collection. If we can create I mean, the NIH is way out there in front in creating these kind of data sets. Now they put everything in the public space, right? All these data, and that's where public money should go. But we need some sequestering so that we can use it in a regulatory environment. And I think that's what would support a lot of use cases that don't get the value right in that one study. They get the value in the aggregation of a data set that is built over time. So that's my gaps.
Oh my gosh, that's fantastic, Brandon. You're giving me some PTSD on figuring out truth and how to adjudicate in the analytical studies that we did. Okay, but that's a good segue, I think, for Susan in terms of Brandon, you're sort of touching on the importance of registries, collecting large sets of data. Can you talk a little bit, Susan, for us about pathology and radiology, particularly in the challenges that might exist, or even the value that we're seeing, but also the challenges that may exist in creating those registries?
Absolutely happy to do so, and super glad to go after Brandon. Maybe we can chat later because you brought up some really good points. So I've been really fortunate to work with the Cancer Institute as well as the CDC and a few of the state registries. I want to just call out Eric Durbin, for example, and the University of Kentucky Network Cancer Registry to the states, and also to the CDER program, the surveillance of cancer. This is just one use case, but I think it will highlight some of the challenges that we're seeing in our electronic healthcare system and the way that we handle data. You know this for sure, that by and large, the pathology reports are completely in a different system, in a PACS system, that are not, at all or very rarely linked to the EHR. We're pulling a lot of data in our side from the EHR and related data. So there's just a real challenge. When you're going to make a new cancer diagnostic, it's the path reports. That's where the cancer diagnostic comes from. When we're looking at that diagnostic being a yes cancer, a certain type, then going to a state registry or to CDER, we're measuring that timeline not in months, but more like 12, 18, 24 months, which is a really hard thing for building research and therapeutics and looking at new diagnostic tools, if that's our time frame. So just shortening that time frame, as well as potentially creating new and innovative tools and AI in that space, is something that's really important. There are two really exciting things that are happening. There's CAP, there's the cancer protocols, a standardized community language that provides standardized reporting that goes up to the state registries. ECAP is the electronic version of that, and getting that implemented in the healthcare system is a real challenge, and something that I'm really excited to work with CDC on. That would really help us enable that feedback loop into the EHR, and that we could then pull for research for CDER and then also for state registries in a more timely fashion. But the adoption of that has been slow. In addition, a completely different technology in the AI-focused space was work that was done with cancer and DOE, Mosaic. It's a hierarchical learning system that was looking to pull at least 4 different elements that could be reported to the cancer registries. I think that they saw good success, about 97% success rate, I think is measured against pathologists actually rating the reports. But the adoption of that is also slow and time-consuming. So there's just a real challenge in integrating the diagnostics of cancer from pathology data back into the research framework or back up into the state registries where CDER will pull their data. We're in a time where we have the capabilities to modernize these systems and modernize how we're doing this. So when we look at registries in general, I think that the high tech and the 21st Century Cures Act has really enabled us to fast forward some of these opportunities, leveraging US CDI Plus and all the variants that I work on, cancer, now cardiovascular, potentially dental, in collaboration with HL7, will help us develop those standardized languages that can be implemented in the healthcare system and then propagated up to the registries in different ways. Also, I'm sort of an innovator of technology, and so one of the things that I work on, and I actually work on this with FNIH, is developing and implementing and deploying innovative tools in electronic healthcare environments that will help us collect and utilize healthcare data for research or for cancer surveillance, for example. So there's a space there for innovators in pathology and other areas to help us streamline some of those processes. And finally, data governance. I bring that up because this is a public-private partnership meeting, and the challenge is, you underestimate the challenge of data governance and public-private partnerships, and that could really use some deep thinking, and I have asked FNIH to help me with that as well, which is how do you look at data governance when the data is coming from stakeholders who are in the private sector, but yet still enabling what we can do in the public sector. So just some thoughts.
That's fantastic. Thank you for that. And thank you for leading the charge and being on sort of the bleeding edge of this. It's so important to have data coming from the wild to help us develop these tools and validate them. Okay, so my last question, and then we can get to some audience questions, is for Jeff. He's been so patient. And so, Jeff, from the perspective of, we know the value of consortia. That's why we are here. And I applaud, I think, Friends of Cancer Research for leaning into some very challenging areas and working with policymakers to try to make a difference. Two questions. One is, you can speak, in your own words, of sort of the value of consortia in moving the needle, which is so important, and many people here, I think, want to move the needle. The value of those consortia and how you think about when they work best. You have a very unique perspective, and what sort of attributes of these consortia we can think about in making them most effective.
Yeah, I think one area of value is identifying areas where one organization can't do it alone. So in most of the examples that have been discussed in the panel, there's multiple different tools that are being developed to achieve some of these common goals, whether it's improving tumor measurement or pathological assessment or, whatever the case may be. So hopefully the consortia role could be to help level set that and do it in a expedited way. I don't want to advocate for half science, but we are in an environment here where the technologies are moving so fast that if the validation process is so slow, it just simply becomes obsolete. And I don't think any of us want to see that occur. So how do we find a sort of an even ground? And the data sharing aspect, I think, is also a unique one. I'm very pleased for our collaborators on different projects. Many of them are here. The willingness to overcome some of these data challenges. I know it's not easy. I've only seen like the tip of the iceberg of what like some of our partners that are donating the imaging for the RESIST project I mentioned, of what they're going through to get it out of their companies. But on the tool developer side, the willingness to also, probably take a little bit of risk by cross-comparing to competitors, so to speak. But I think that just speaks to the scientific motivation of this. These are scientific projects, and I hope that these consortia-type projects, yes, you want to see them ultimately achieve the validation in many cases that they set out to do, but there's still learnings here, even from a proof of concept or thinking through the precursors of what validation could look like, or identifying different scientific opportunities, or developing the reference sets that might be the basis for validation into the future. I don't think from an endpoint or biomarker standpoint, you may not always want just one company doing that. If it has broad applicability, having the consortia to be able to bring together various stakeholders, I think, is really important.
Fantastic. Okay, people at the mic have been so patient, so thank you for your patience. We certainly have time for questions, so I invite people to come up to the mic. So we'll start over here, perhaps.
Thank you. I'm Michelle Long. I'm with Novo Nordisk, and I'm in drug development, and I work in the space of fatty liver disease or steatotic liver disease. And for this condition, you use pathology to not only identify the patients for your clinical trial, but it's also your endpoint that we use for accelerated approval and then ultimate approval in the non-cirrhosis space. There's so much really great discussion and talks, and I want to just talk a little bit about the human factor here. Some of the things that we face in a MASH context trials that you have patients who have a biopsy done for clinical purposes. They could submit it for central read, and you would think that they would look and see, okay, is this person likely to be enrolled in the study before submitting it? Well, in reality, we see that 60% of those patients actually screen fail from the study. So it already starts making you wonder who's setting the truth here. Certainly not your local community pathologist. And then we also know that you ask the same pathologist to reread the slide in a different context, and you might get a different answer, even for things such as F stage or fibrosis stage, where really, it makes sense that you should be able to trust that. And we've kind of built our whole discipline of hepatology around, that sort of trust that's there. So I really like the guidance that came out also from the FDA to say, okay, it really depends on the context of what is the decision, how is this AI-based tool going to be used? And it makes sense that, if it's going to be something that is a regulatory endpoint, has to have a different standard, total sense. But in my mind, how do you play the human factor here? Because at some point, it's actually the human factor that could be the liability. And so it's almost like, if you want to have a reproducible standard, then you want to actually limit the human's ability to manipulate that standard. Whereas I think some of the way that it's phrased, it's almost like the more important the decision, or if it's an endpoint for regulatory decision, I'm kind of hearing a tone of, okay, maybe the human should be able to overrule. But in some sense, you could argue for the exact opposite, just to maybe a little provocative, but curious your thoughts.
Yeah, I think that last thing you said is super important. I'm going to see who wants to address this. I think, Jeff, you showed some data in the work that you guys did at Forensic Cancer Research, how the tools themselves actually can be variable, which is interesting. Everybody sort of thinks, well, AI is going to solve for all these problems about variability, but in fact, 10 different AI tools are as variable as 3 different pathologists are. So there has to be a way to think about how to have humans in the loop enough, but still extract, I think, the value that AI is providing, which is a little bit what you're getting at. I mean, who wants to tackle that? Yeah.
I mean, I can quickly say what we did. I mean, we very much saw this problem in how we designed our clinical validation study for our AIM-MASH tool. And first is there's analytic validation, where the tool operates autonomously, essentially. And the ground truth, unless you can go to outcomes, which is often very hard. And as I mentioned here, we wanted to measure an already established set of features. We had to use a consensus of experts and really make that the ground truth. And then for analytic validation, show that the model is non-inferior to any individual human against that ground truth. You might say that's sort of biased against the algorithm, to Michelle's question, which it is, but still, that's the standard we have to apply to it. And then when we did the clinical validation, there's 2 big sources of errors, and we focus a lot on accuracy, and that's where we're saying non-inferior to a human in terms of accuracy. But noise or lack of variability, to me, that's the biggest hidden thing, and I think Brandon mentioned this, that although the 10 different algorithms were somewhat different, they're essentially all deterministic. So if you ran any of them on 2 different times, you'd get the exact same answer. And so they're very good at minimizing noise. And that's one of the huge complementary advantages of a machine learning-based system, is the fact that it's deterministic. We have trouble trusting large language models and generative models because the same input, the same prompt doesn't always produce the same output. Guess what? What we're trusting with pathologists is very much the same. They're generative models in your head where the same prompt, as we've heard many times, at different times of day or different days of the week, will produce different outputs some proportion of the time. So the system, the deterministic part of the system, is actually super valuable. And the way to not dilute that too much is to have a system that's both very accurate, and in our case, when we designed our clinical validation for AIM-MASH, it was all designed around what we called a 2-point rule, that a pathologist could only change the score if they thought it was really not just a little bit off, but way off. And that rule, worked for us in the study in the sense that we were superior for 2 of the most important measurements, ballooning and inflammation, and we were superior to the human ground truth. And it was obviously, and we were non-inferior for everything else. But it was way more reproducible. So you kind of want to be non-inferior or superior on accuracy, but at least not inferior. And you're going to be far superior reproducibility and lack of noise. So that's how we designed that for that reason. Once you let someone change it for just small tweaks that they disagree with, you lose a lot of the benefit of the tremendous reproducibility of the system, which is particularly important in areas where it's being used when you're doing post-treatment. The difference between post-treatment and pre-treatment, you really want to minimize that noise because there may not be a platonic correct answer. It's just you want to be measuring the same way after treatment and before treatment, and then across a cohort of patients, you expect the drug will be more effective for measuring that change than the comparator arm, in this case, usually placebo.
Awesome. Okay. We'll head over here. I just want to underscore the 2-point rule. It was a little controversial with the agency. There's public information about that I would encourage people to read, but it's a very important point. And I think as we think about validating AI-based tools for radiology and pathology, that 2-point rule didn't set a precedent, but it set an interesting concept of how much disagreement should be allowed in the validation. So it's something that we can talk about in the future. We'll go down there. Yeah.
Suzanne Wayne, I'm from Novartis. In the morning when I was doing the quick comments, I didn't introduce myself. So AI is a super powerful tool. As a AI enthusiast myself, as a drug developer for 20 years, I think the potential of AI in the imaging analysis is way more than we currently can imagine. That's number one. So more importantly, I want to emphasize the importance of training and the validation. So in the past 20 years as a drug developer, I haven't spent 2 years in an imaging company, so clinical trial imaging company. And that time I was the bridge between the drug developer, myself as a medical director at the time, and then the dozen of expert readers. And there's a very rigorous way of choosing the expert reader because those experts in different, someone is really good at RA, someone really good at fracture, and then you choose the best reader and you choose 2 out of maybe 6 candidates and make sure you validate them and cross-validation. It's a very rigorous way of doing that. And I think for AI, we should have even equal or more rigorous way of validating. Therefore, the training of the AI, I happen to be able to work on one project 3 or 4 years ago, is the early stage of AI training, reading the sacroiliac joint. And then when I was asking who trained those AI to define, to delineate the sacroiliac joint, because me, not the expert reader, I was thinking that doesn't look right. And they said, oh, our radiologist. I said, is that radiologist ever an expert reader and read like tens of thousands images? They said no. So therefore, I think one point really important is when FDA or our regulatory agencies to define which AI tool being approved, this training and validation is super important. It's not every AI, when they come with the good-looking graphs, it's a good tool.
Absolutely. Yeah. Training, no doubt. I think the agency places a high premium on understanding, right? Would I be correct in saying that? On understanding how training data is included. Yeah.
Maybe I can add a little bit more. I think both training data and testing data are very, very important. In my personal opinion, I think the quality of the testing data is even more important than the training data. Because sometimes for training data, it's okay to actually sometimes get some data of various qualities and even sometimes outside the scope of intended population, because sometimes you can borrow information from other type of data. But it's really, really important to maintain high quality representative data for the testing sets.
Excellent. Okay, and over here.
Brandon, I have always considered myself to be a noisy version of the truth, so it resonates with me. My name is Mike Espy, and I'm with the NCI, and thanks, Susan, for the shout-out on Primed AI. I'm interested in the title of the forum today, Clinical Imaging and Pathology Data. So we haven't talked about the and at all, really. So what is the state of the field in terms of multiscale? So we have image-guided biopsy, but At what point does a biopsy inform the radiological imaging interpretation? Can we move across scales? And can we connect the world of pathology and radiology in interesting ways by virtue of AI?
Anyone want to tackle that? Yeah, go ahead, Andy.
Sure. I think on the discovery side, there's a lot of work going on. Putting together those data sets is still really challenging. Part of it's that most of pathology historically hasn't been digital. So we're still in, I'd say, the rapid adoption phase of digital, whereas radiology is there. So there aren't great registered data sets at scale. So there's a lot of manual registration for areas where looking at both like prostate or bone are used, but it's not yet systematically aligned with large-scale data sets for machine learning. And I think we're headed that direction, but I do think it's largely sort of a clinical integration and infrastructure for building these really rich data sets we're still in the early stages of. And then, of course, the really killer use case, of which I think there are multiple, but I still think it's in the R&D phase, and clinically we're not yet integrated enough to have the impact shown.
Great, thanks. Hey, Josh Vessel, Diana Health. We've heard some great discussion here about the human in the loop, and I want to think about with you the right places to bring the human into the loop. And I'm specifically thinking way back at the beginning of when you're developing an AI-enabled tool. There was a really cool paper put into medRxiv about 3 weeks ago about using human-centered design in clinical predictive AI algorithms. And the point there was that if you build this beautiful thing that works in silico, but you don't think about who's actually going to use it, it's never going to get used. And I had a discussion on social media with a friend of mine. I said, this is the chocolate hammer problem. A chocolate hammer is a really cool tool, but nobody actually needs a chocolate hammer. And no one's going to use a chocolate hammer. And we shouldn't build chocolate hammers. And so as we think about what we're aligning to, whether it's getting data standards right up front to drive AI, tool design, validation, and testing, if we think about aligning to regulatory standards, how do we keep the end users, plural, in mind from the beginning so we don't build chocolate hammers?
I don't know if anybody wants to tackle that. Yeah.
Yeah, maybe I can tackle that. And I have to say, we've been talking with the Society of Ergonomics about this whole concept of developing human in the loop at the very beginning in terms of developing algorithms. I didn't talk about a program that we support, Aim Ahead, which is essentially exactly that. It's not only developing capacity and capabilities of AI across the country, mostly looking at under-resourced institutions and centers, but it's also about bringing in clinicians from rural clinics or public health sector to help define what AI tools we're going to develop in that program, and then working with us as we develop these AI tools. This has been transformative for the AI researchers because, exactly as you said, oftentimes they think what the clinicians need is another really cool widget that will help them diagnose patients quicker. But what the clinicians actually say is, I really need some AI tools to help me understand why my patients aren't actually coming to the clinic. And so it just flips the dynamic completely on its head, and we still are working to develop really innovative tools, but not necessarily always in the innovation of technology space in the sense of new FDA-approved devices, but more on the ground AI capabilities that really serve a purpose for a particular community. And so I completely take what you're saying as an important point, and thank you for bringing it up.
Mike? Yep, Brandon.
I wanted to say that, my research has involved a lot of volunteers. I have not done the calculation of how many hours of pathologist reads I have inert in my data that I've collected, but it is probably at least 100 and probably more than 500. I don't know the numbers, but the key is there is a hunger for understanding AI. There's a hunger for understanding the clinical interpretation process and CME credits and how to do this from an interpretation level. That's how I got people in. I'm a mathematician, but the project I ran created a CME course because of my collaborators, because people wanted the tools for interpretation. Why did I want it? I wanted to reduce the variability in my reference standard reads. And so it served both of those purposes. It doesn't have to be all locked into one funder paying for everything if you open it up in terms of improving the clinical space.
Great point. Okay, yeah.
Hi, it's Joe Manetsky, FNIH. We heard today from you, AI in mucosal healing, AI in cancer, AI in various other pathologies, and that's great. I mean, it's a great way we could be doing things, but I'm wondering, we like to get similar minds together, and right now all of the AI, kind of if there's a vendor, is kind of aligned with a disease. And I'm wondering, from an FNIH point of view, are there opportunities where we can get multiple AI companies together to work in a pre-competitive space that we could help with, right? So that's kind of, and Susan mentioned data governance. That maybe she offered that one up, but are there other things like that? Because it would be, in a pre-competitive space, that might be an opportunity for us to step in and, not raise all boats, right?
Yeah, I think it's a great point. I think sort of the difference between point solutions as we think about an AI, which is a very specific use case, which 2 years ago, not even, maybe a year ago, was more relevant, whereas now we're moving into multimodal, multi-imaging, multi-modes, and building models against that is now becoming much more relevant. It's really the state of the game. So I'm interested in anyone's thoughts here on the panel, but certainly I think to the extent that multimodal models now are becoming a very important aspect of AI and ML, I think the FNIH can play a role in bringing together disparate, so to speak, point solution providers and seeing whether or not they can collaborate on multimodal model generation. That's the thought. Yeah. Yep. Excellent.
Hello, Dr. Montalto.
Hello, Dr. Townsley.
This is my boss, by the way. So this is for all of you. I mean, I could talk about this forever. I just find this so fascinating because it really does feel like the future, right? And in 20 years from now, we're going to be like, remember when we were talking about this? I just wonder, when we're thinking about AI and coming back to this idea of risk, and the potential risk in terms of outcomes, getting it wrong, are we adequately assessing potential for benefit too? Right? I mean, I'm a doctor at the end of the day. It's a risk-benefit relationship. Everything's a risk-benefit deal. And I do wonder if we are adequately capturing that. I wonder if we're adequately capturing the potential for benefit for patients to maybe get shuttled to the right trial and get shuttled to the right screening program. I wonder if we're capturing whether or not patients are spending their time getting screened for multiple trials versus actually getting started on therapy, which is what they want. And so given that, I just wonder, again, if there's opportunities for us to actually embed quality of life measures or really think out of the box about how this can help. I think when I look at this, I think of sitting with a pathologist. I'm a hematologist. Sitting with a hematopathologist, looking at the bone marrow and helping that pathologist think about what are the stains they should do next based on clinically what I've been seeing at the bedside. And so I can't help but think that AI is somehow going to be a tool in that way. And we need to talk to the pathologists, talk to the clinicians and ask them what is it they want, because ultimately they're not going to accept things that we force upon them. It just doesn't work like that, right? New technology doesn't work like that. So, but ultimately they're going to say, hey, I want this. And I think Andy, you're already hearing that. Some academic centers are like, we need this. We want this, right?
Yeah, totally. So maybe just, really quickly, there's this interesting, sort of speed of or sequence of how things change, where you might think everything happens in drug development that happens in clinical, but at least in drug development, typically they want to innovate on the drug and everything else is kind of standard of care, and why rock the boat? So it took there was sort of a high activation energy, I think, for it to become really ubiquitous on both sides. But I would just say we're absolutely seeing it on the diagnostic side, driven for internal reasons, as you mentioned, driven for wanting to be more efficient, wanting to be supportive, wanting the best platforms to share images with other experts around the world. So one stat that came out this year was about 50% of labs saying they're using digital pathology in some aspects of their clinical work today, and then an additional 30% saying they're going to be doing it in the next 12 months, and only 20% saying, you know, this is maybe in 2 years we'll think about it. And that's just a huge change over the last 10 years. So we are very much in a rapid adoption part of that phase. And to your point, it's been driven largely from the medical community itself and not from pharma sort of pushing it.
Yeah, I think we're kind of up on time, if I'm right. I think we're heading into the very last section, which are going to be some awards. So, Chief, it's okay, maybe we can cover it offline, but I want to thank the panelists. I want to thank the audience for such an engaging discussion. Thank you so much.